Hello everyone, my name is Suzanne Alvernaz and I'm a third-year bioinformatics PhD candidate at the University of Illinois, Chicago. I'm very excited to present our work on creating a user-friendly pipeline for longitudinal microbiome network analysis, LIMÓN. Existing methods for identifying temporal alterations in microbial communities are limited. LIMÓN focuses on overcoming these challenges by accounting for overdispersion, zero-inflated count data, compositionality, sample covariates over time, repeated measuring errors, and individual network characteristics. LIMÓN is a robust platform for detecting longitudinal changes in microbial interactions over time, allowing for the identification of relationships between network centralities in a sample feature of interest. LIMÓN's approach involves three key steps. This package is designed to take microbial count table and it provides sample metadata. The format is required to have a column of subject IDs, the repeated measure, and a column of time points. Users can also provide covariates of interest. The count data are fit to a zero-inflated negative binomial linear mixed model with each ASV as an outcome and the covariates as predictors. The corrected values are extracted by subtracting the residuals from the fitted values and splitting by time point. Users can specify if providing raw counts or proportion data of relative abundance. A network per time point is then inferred with the covariate corrected counts using Speakeasy network inference. Users can pass the usual arguments to Speakeasy for parameterization. Note, this function performs CLR transformation for data normalization. The edge table and iGraph networks are extracted from this output for users to transfer to Cytoscape for better visualization. Individual sample networks are then inferred using the time point specific networks. For each time point, the algorithm will iteratively remove one sample and infer a new network for that time point. The weighted difference between the network for all samples at time point n and the I-j network is the network for the jth sample. The algorithm also provides users with options to extract the edge table per sample here as well. The final steps in the pipeline provide users with the option to extract a set of network centrality measures as well as an interaction/edge weight table to add to their sample data for statistical inference. This is an example of a pseudo-example of the LIMÓN utility in longitudinal microbiome analysis. A. Network definitions, an example of the type of network centrality measures estimated per sample in LIMÓN. These include measures such as closeness, betweenness, and number of communities. Edge weights between nodes, or ASVs, represent the strength of interactions. B. Network characteristics over time by group. Centrality estimates for each individual network can be extracted from LIMÓN outputs and used to test network characteristic alterations over time. In this pseudo case, the number of communities is compared between two treatment groups. C. Microbial interactions over time. Similar to B, edge weights, or the interaction between ASV pairs, can be examined over time by group to identify differences in community connections. Our ongoing work includes applying LIMÓN to a longitudinal infant microbiome dataset. Data were extracted from Ohm et al. 2019. From these data, we pulled 3 fecal sample time points from 86 preterm infants at around 10, 20, and 30 days of life. 24 of these infants developed necrotizing enterocolitis, a dangerous gastrointestinal infection, within the first month of life. The fecal samples were sent for metagenomic sequencing. We are currently using LIMÓN to examine longitudinal differences in the gut microbiome development between healthy and sick infants. Please refer to these references for more explanation of the development of the LIMÓN methods. And LIMÓN is an open source package that can be downloaded from GitHub. Please see the tutorial for more information and don't hesitate to reach out with any questions or suggestions. Thank you.